Hello again, and welcome to Biocytogen's webinar series. It's nice to meet you all online. It looks like we are ready to begin, so let's get started. My name is Xiaoxuan, and I'm the marketing specialist at Biocytogen Beijing, and I'll be serving as your moderator for today's session. So in today's session, it is titled RenNano Heavy Chain Only Antibodies Powering BBB Crossing, Multispecific Antibodies, ADCs, and In Vivo CAR T. So into today's sessions, we will be exploring how heavy chain only antibodies and nanobody-based approaches are enabling sort of a wide range of next generation modalities. It is my great honor to introduce today's speaker, Dr. Frank N. Dr. N is the Vice President of the Institute of Biologics, which is Biocytogen's Antibody Discovery Division. His key responsibilities include overseeing Biocytogen's internal and external collaborative antibody discovery programs, covering oncology, autoimmune disorders, rare diseases, and more, and including those that have reached PCC stage, non-clinical studies, IND, and clinical trial stages. He is also in charge of portfolio strategies, target inception, and product profile conceptualization, as well as overall project management and implementation. Frank earned his PhD in cellular and molecular biology from Brandeis University, followed by a postdoc fellowship in immunology at Dana-Farber Cancer Institute and Harvard Medical School. With over 20 years of experience in drug discovery across industry and academia, Frank took increasing responsibilities before joining Biocytogen, including positions at Bicycle Therapeutics, Horizon Discovery, Millennium Pharmaceuticals, which is now known as Takeda, and Broad Institute of Harvard and MIT. In today's talk, Frank will share how Biocytogen RenNano platform is enabling the discovery of fully human, heavy chain only antibodies and
Thanks, Julian, for the nice introduction. And, hello, everyone, and welcome to our webinar. So today we're going to talk about Biocytogen's RenNano heavy chain only antibodies platforms. So my talk, hold on a second. Trying to advance the slides here. Okay, so my talk is going to be in three parts. The first part is a brief introduction of what is the heavy chain only antibody, or called various names, VHH, single domain antibodies, nanobodies, or nano antibodies, and they all mean the same thing in this talk. But we're trying to consolidate the names to a couple, like heavy chain only antibodies or VHH. So first is a brief introduction of what that format of antibody is, and then I'll do a brief introduction about Biocytogen's specific heavy chain only antibody platform, and then I'll talk about some case studies, applications of heavy chain only antibodies. First is the introduction of the heavy chain only antibody. So before 1989, this is what our knowledge about what an antibody is like. So this is a conventional antibody that is comprised of a heavy chain, which is the blue part, that comprised of VH domain, which is the binding domain, and a constant regions, CH1, CH2, and CH3. So this is the heavy chain paired with the light chain, which are the two green domains, the variable light chain domain and the constant light chain domain. So this is half of the molecule. The conventional antibody is a dimer, homodimer. So there's a bivalent, there's another copy of everything on the left side. So this is the conventional structure of antibody, and you can also take the binding domains, the variable regions of heavy chain and the light chain out and join them with a linker to stabilize the structure. So we call that a single chain variable fragment, or short for SCFV. So this is the smallest binding units for this conventional antibody. We call that fragment of antibody binding, or short for FAB. In 1989, a team, Professor Hammers, and his team uh, discovered another form, a smaller sized form of antibody from camels or llamas. Okay. And the difference is that this antibody only has the heavy chain portion. So if you look at the conventional antibody, it contains only the blue part, the VH domain, and a constant region that comprised only of CH2 and CH3 without CH1. So this is what this antibody is like. Here's a binding domain, the heavy chain only, without the light chain at all. But this molecule, as shown, carries the FC region, which comprised of CH2 and CH3. And the minimum binding domain, if you take this blue domain out here in the ribbon structure, and this is the smallest binding unit. It's only 12 to 15 kilodalton. And so this is the binding unit. And it's also called the variable heavy domain of the heavy chain, short for VHH. So after 1990s, so we have an additional format of antibodies in addition to the conventional IgG format antibodies. Here lists some of the major differences or differential points between the conventional IgGs and the VHH. So that is first is very obvious is the 10 times difference of sizes, and also something associated with sizes like tissue penetration faster with the VHS as it's much smaller, and also the size of the paratopes and some biophysical properties. And then I just wanted to point out that traditionally the IgGs, conventional IgGs, have been dosed usually through IV or SC. However, some innovative formulations of delivery route now for antibodies includes inhaled formulation and oral delivery. And the VHH format is, particularly good for inhaled and oral for formulations because of its small sizes and high stability. So here lists a few of the approved marketed products that comprised of the heavy chain only binding domains. This is anti-VW factor antibody approved for a rare blood disorder. It's anti-clogging function. And here is anti-PD-1 VHH antibody for immuno-oncology. Here's anti-TNF alpha nanobody. It's actually multispecific nanobody that targets TNF alpha and human serum albumin. So, and the next one is actually a CAR T product, cilta-cel, that is approved to treat multiple myeloma, and the binding domain are two different VHS binders for different epitopes on BCMA. And the last one is not approved yet, but in 2 phase 3 studies is a multispecific VHS antibody that targets IL-17A and F, and also the anti-human albumin domain. This is to extend the PK or half-life of this antibody. So this heavy chain only antibody is the most versatile building blocks, okay, for making for a lot of therapeutic uses. For example, for making bispecific or multispecific antibodies, because it contains only the heavy chain, it can pair with any other formats, antibody formats, including itself, for example, or the FAB formats in this 2+2 bispecific antibody. So it also, because of a smaller size, it's liked by a certain type of antibody drug conjugates, for example, the radiation therapy, targeted radiation therapy. And we talked about for some new popular formulations of antibodies for specific indications, the oral form and the inhaled form. And also, the last but not the least is for in vivo CAR Ts, both as the targeting moiety on the, for example, here is LNP particles, but also the CAR part. Okay, here's the mRNA of CARs, and the CARs are the targeting moiety. You also, people like to use the VHH heavy chain only antibody as the binding moiety because of the small size. So we'll talk about all these things towards the third part of the talk. Okay, now let's see. Who we are. What is Biocytogen and its heavy chain only antibody platforms. So Biocytogen is a global biotech company headquartered in Beijing, China, with the largest animal facility in Asia in Jiangsu province. And we also have close to 150 people strong team in Boston, and with the business offices in Shanghai, China, San Diego, and San Francisco in the west coast of the United States, and also Heidelberg, Germany. So in terms of the company, the work is divided into two major parts. One is antibody therapeutics that is responsible for antibody discovery. And another arm is preclinical product and services that are responsible for our 5,000 more different animal strains, cell models, and pharmacology services. And at Biocytogen, we work on these different modalities that includes monoclonal antibodies, and bispecific or multispecific antibodies, and derived from this platform, the bispecific and the multispecific ADCs. And today's our focus is on the heavy chain only antibody platform. And we also have a TCR, therapeutic TCR platform through which you can identify novel TCRs for intracellular targets. We also have two programs for hard to hit in terms of antibody discovery classes of molecules. One is the peptide and MHC complex that's called a TCR mimic antibody platform, and also for the 7' transmembrane GPCR molecules, the GPCR platform. So here are the three fully human transgenic mouse strains, collectively called the red mice, that are the working horse for our antibody discovery, fully human antibody discovery at the Biocytogen. So the first mouse strain was called the RenMab. Ren in Chinese, it means human. MAB stands for monoclonal antibodies. So this is the most diverse fully human antibody transgenic strain that carries the entire human heavy chain repertoire and the entire human kappa light chain repertoire. So that's to generate monoclonal antibodies with the most diversity. The next mouse strain, transgenic strain, is called a RenLight. Ren again stands for human, and the light stands for common light chain. So the difference between RenLight and the RenMab is actually on the light chain part. The heavy chains were constructed the same way. On the RenLight platform, a single light chain is in place of the entire human kappa locus, so that all the antibodies will carry the same common light chain. So this makes making bispecific and multispecific FAB format antibodies are really an easy task because you don't have to worry about the mispairing of different heavy chains and light chains. And then the last is the RenNano, fully human transgenic mice. And on the next slide, I'll describe how this mice was constructed. But as we said, that it's a very versatile format to make bispecific and multispecific antibodies. So we have licensed our RenMice platform to 50 different partners, and and we're also very active in helping people other than Biocytogen using these mice for antibody discovery. So this slide shows the construction of RenNano or the heavy chain only antibody strain. So here it used to be the mouse heavy chain locus, and we used homologous recombination, and in situ in one step we replaced the mouse heavy chain locus with the entire human heavy chain locus. And then also in this mouse strain, we removed the light chains, certain light chains, and also we retained the mouse constant region, except with the modification that we removed the CH1 domains from those constant regions. So the resulting, in the resulting RenNano, Ren again for human, nano is for nanobody. So in this mouse strain, we have the human variable, the heavy chain variable region connected to the mouse CH2 and the CH3 domains. And so how do these heavy chain only antibodies from Biocytogen, perform? How are they like and how do they perform? So here it shows that the diversity, these antibodies, the heavy chain only antibodies actually have very good diversity. So here is the representative of 5 different campaigns for nano heavy chain only antibodies. On the left are panels of the germline gene usage, and you can see that it uses the common IGHV 3 and 4 genes, but also some rare ones like gene family 6 and 5 here. So that's germline gene usage, and also in terms of the CDR3 lengths. And you can see there's a wide distribution of the lengths, which indicates diversity, and a significant portion of the CDR3s also is longer than the Euro median between 13 and 14 amino acids. So it's the HCAbs from Biocytogen can be very diverse. And here's a binding characteristics in terms of affinity. And here is that shows the binding affinities. Here's the lines, binding affinities for nanomolar potency, and 10 nanomolar affinity. So you can see a significant portion of the cones from 8 different campaigns, and binds between 10 to the minus 8 and 10 to the minus 9 molar affinity, which is a typical range for the conventional IgG binding. So in terms of even a couple have sub-nanomolar potency. So you can see these heavy chain only antibodies behave just like the conventional IgG antibodies, the FAB format. And this slide shows the excellent, developability of our heavy chain only antibodies. You can purify them. It's a lot easier if you have FC to purify them to high purity, and also in terms of biophysical properties, low hydrophobicity, lack of aggregation propensity, and also in terms of thermal stability and have excellent TM values. So these are all very important properties to make antibody drug. Here's a list, a partial list of some of the campaigns that we ran on the RenNano mice to generate heavy chain only antibodies. That's to give you a flavor. We have a lot of targets in oncology, and we should have autoimmune diseases, and also neuro for neuro and metabolism. So we actually, yeah, the Millennium, we have a large antibody library that the heavy chain only antibody is an integral part of this large library that has over 1 million clones. And for HC antibodies, I think we have at least tens of thousands of clones that can be used. And also, we're interested in seeking collaborations with any of you so that we can explore these valuable assets together. In terms of how we do generate the antibodies, one thing I wanted to point out is that not only we have the Ren mice, Ren mab, Ren light, and Ren nano in wild type form, but we also have the knockout. In other words, we knock out the target genes on the background of RenMab, RenLight, and RenNano. So we do that to break the immune tolerance against the highly conserved regions between mouse and human targets, so that the targets would be entirely novel to the mice when you do immunizations. And, and also the Biocytogen, we do from end to end in terms of antibody discovery. So our teams can do antibody discovery in vitro, in vivo characterizations, push the binders all the way to PCC, and then we have a very capable CMC team that will be that can do a cell line development and upstream and downstream and formulation development and work with the CDMO to deliver the entire CMC project. And also, we can perform together with the CRO, our partner CROs, or the NHP tox studies and the IND enabling studies. So we provide end-to-end services and also collaborations with our partners. Okay, the third part, vignettes of HCAb or heavy chain only antibody applications. So I'm going to talk through a number of examples where we used the heavy chain only antibodies. So the first is heavy chain only antibodies for blood-brain barrier shuttles. So we all know that the brain, in order to protect its delicate content, will not have materials inside the brain just on a random basis. So there's this brain endothelial cells has a very unique property. It's called the blood-brain barrier that would prevent a lot of large molecules, for example, antibodies, even the smallest antibodies like VHH or heavy chain only antibodies from entering the brain from blood vessels. So in order to deliver drugs into the brain, so people came up with ideas where they would trick the system by using binders against some endothelial cell targets. Most popular ones are TFR1 or transferrin receptor 1, also called CD71. And another target is CD98, which is the heavy chain of some amino acid transporters. So by using binders targeting these two targets, the binders can be fused to therapeutic antibodies, and in this way, you can bring the cargo into the brain from the blood. So those kind of oranges blocks are called BBB shuttles. And this slide shows our TFR1 based heavy chain antibodies serving as BBB shuttles. So we ran a campaign. We identified a number of clones, and here is the in vivo characterization of the brain penetration. So if you focus on the middle panel, parenchyma components, so you look at the blue line is the benchmark positive control, which is a TFR1 shuttle approved for a rare genetic disease for enzyme replacement therapy, JR141. So that's the blue line. And the gray line is the isotype control. And the red, purple, green, and black represents our TFR1 BBB shuttles. And you can see that a few of them have more brain exposure than the benchmarks. Okay, so that's the structure of these brain shuttles that we tested in this study. And we also did more characterizations. So we made a monovalent form, and we showed that the monovalent form of one of the antibodies, AB38, and another one, AB26. For both clones, the monovalent form outperformed the bivalent form, which is consistent with this class of TFR1 based BBB shuttles. And here is another BBB shuttle targeting CD98, and here we have 1, 2, 3, 4, four RenNano or heavy chain only antibody based on CD98 brain shuttles. We compare that with a benchmark CD98 antibody, which is a monovalent form in FAB format. That's in red here, and the isotype control here is in blue, and you can see all of these RenNano based CD98 shuttles have more extreme exposure than the benchmark. Okay, and we also have a RenLight called RenLight 12, number 12, based on the RenLight format that was also tested here, and it also showed a very good brain exposure. So without getting through all the data, I just wanted to say that our top RenNano and RenLight CD98 based brain shuttles are very good in terms of their in vivo activities. Brain penetrations are much better than the benchmark CD98 brain shuttles. Okay, so the next example is heavy chain only antibodies for bispecific and trispecific antibodies. So the first example is BCG36, which is an anti-PD-1 and anti-VEGF bispecific antibody in this 2+2 format, with the anti-VEGF-A being in the nanobody format. Okay, and here's the data that shows that the PD-1 arm it actually is constructed with Keytruda, and in this bispecific antibody format, it shows the same binding properties like Keytruda as expected, but much better than the benchmark bispecific antibody. This is Summit antibody that has been making news in terms of this bispecific targeting. And here's the VHF arm, and the blue one is BCG38, our own RenNano anti-VEGF binding unit, and you can see it binds much better versus these two benchmarks of anti-VEGFs. One is from the ivonescimab, And another one is from the bevacizumab, which is the classical anti-VHF molecule. So our binding is much more potent than these two benchmarks. And also here we show that our BCG 36 outperforms the ivonescimab in this CDX model. Ours is the red line, and the benchmark is the green line here. And we also show that our molecule has a better PK because this benchmark molecule has a faster half-life, and ours has a much extended half-life in humanized FcRn mice. This is another trispecific. This is actually a trispecific molecule with a nanobody moiety. This is DLL3, CD3, and 4-1BB T-cell engager. All is a simple, bispecific T-cell engager plus the secondary co-stimulatory signal arrived from 4-1BB moiety. So that moiety is a RenNano heavy chain only antibody. So here are some of the data about the performance of this trispecific antibody. Here is the proliferation. Actually, you can see that compared to bispecific antibody, a T-cell engager, ours can stimulate T-cells into more proliferation than the bispecific format. And this is the tumor volume in Vivo on the last day of the study. And you can see that our trispecific molecule, reduced the tumor volume more than the bispecific molecule without this 4-1BB nanobody moiety. And when you do check the activities of the human CD3, the activated T cells and trispecific antibodies stimulate most of the humanized CD3 positive T cells in mice. Actually, a lot more than our benchmark bispecific TCE. This is CXCL10 is another biomarker for the activation of immune systems in this humanized mice, where we tested the trispecific TCEs, and you can see that again trispecific antibodies stimulated more the immune responses than the bispecifics, and also the benchmark bispecific T-cell engagers. So that indicates our, the 4-1BB nanobodies works really well in this trispecific format. This is again another trispecific format targeting different set of targets, EGFR, VHF, and the CD98 heavy chain. And here the CD98 heavy chain was not used for the brain shuttle, but we attach this to this bispecific antibody in order to improve the target internalization. So here's the structure of this trispecific antibody, and all the binding moieties are composed of heavy chain only antibodies. And without going into the details, just look at the BCG38 is represented by this orange line here in 2 PDX models, and our trispecific antibodies outperformed their parental antibodies, 3 parental antibodies, and also benchmark anti-EGFR antibodies and VEGF antibodies. And so the trispecific molecule really worked well in vivo. Okay, now we switch to another set of applications, heavy chain only antibodies for ADCs. And here is anti-FOLR1, heavy chain only ADC for ovarian cancer. And the molecule is called BC. Okay, it doesn't have a BCG name. We just use the anti-4R1 ADC, which is the red line, the most potent one, compared to two benchmark anti-4R1 antibodies, which the top one, the orange one, actually is approved product for ovarian cancer. So this is a PDX model with moderate expression levels of 4R1. And we also tested that in the 4R1 low expression PDX model. And you can see in both scenarios, our molecule, which is represented by the red curve, was the most potent one relative to the two benchmark molecules, ADCs. This is another DL3 targeting heavy chain only ADC for small cell lung cancer and neuroendocrine tumors. And it has very good binding, two clones, AB10 and AB11, and have very good binding of affinities. And in these two CDX models for small cell lung cancer, and our two ADCs, ADC10 and ADC11, the black and orange curves, outperform the benchmark DLL3 ADCs, including the Zai Lab's ZL1310, that actually showed a very good efficacy in human clinical trials. Okay, now here's the last application of heavy chain only antibodies is for in vivo CAR T. In vivo CAR T has become a really popular concept in the last a year and a half or so, and this culminated in recent news in April, where Eli Lilly paid $3.25 billion for a in vivo CAR T product from Colonia, based on only phase 1 data from only 4 patients. So it's a very hot topic, and in this year's American Society of Gene and Cell Therapy conference in Boston, actually earlier in May, in vivo CAR T is said to be everywhere. So it's a very hot topic. So it's hot because in vivo CAR T you can overcome almost all the major shortcomings of traditional CAR T. In terms of you don't have to do lymphodepletion, you don't have to collect patient cells and grow ex vivo of those patient cells, which is a very costly and time-consuming process. So here you only need, for example, to deliver the CAR construct to the T cells inside the patient, and then you can get the CAR expressed, and those T cells will be live CAR T cells and attack the tumors. Okay, in order for this to work, okay, first you need one way to direct these LNP particles specifically to T cells to avoid a non-specific the delivery or side effect associated with that. So that's one requirement. And second is that probably the CAR molecule here is depicted as a ICFV, but if you can have a heavy chain only binders here, that actually would make the construct even more compact, and that would help the expression levels. So one is targeting and another is expression. And the molecule we're talking about is a CD7. So we generated a heavy chain only binders against the CD7 and to be used as the targeting moiety for the LNP particles. Okay, why CD7? Because CD7 is actually the earliest T cell lineage marker expressed on pro-T lymphocytes. So that's the earliest form in T cell development. And it expressed uniformly at a high level in mature T cells. So this makes CD7 a really attractive target to target to generate antibodies so that the antibodies can direct LNPs specifically into T cells. So here is some of our data about our CD7 program, heavy chain only antibody program. Here's a number of clones, and we have a positive controls. This is internalization and activities, and you look at the affinity. The positive control of benchmark molecule is in red, and we do have a few clones with affinities right in the ballpark of this positive control in terms of human CD7 binding and also in terms of the monkey CD7 binding, although the monkeys shows a little bit of weaker affinities. And here is the functional test in vitro. The red and the green are two clones, RN11 and RN24 of our heavy chain only antibodies against the human CD7. And the brown marker is for the positive control or the benchmark. And you can see one molecule performed similarly in vitro in terms of delivery content cargo, which is a GFP molecule into T cells, and also in vivo, and the RN11 anti-CD7 molecule also, delivered the LNP molecules in vivo to a similar level of that benchmark molecule. Okay, so with this, that was all the some of the selected applications that I wanted to share with you today. And to wrap up this webinar, I just wanted to point out that we have a very active business development team, and we're very open to work with anyone on our antibody assets that includes these heavy chain only antibodies. This is a partial list of the business deals we had with the big farmers and biotech families alike. And please feel free, if you have any questions about these binders or anything, please contact our business development team, and they will help you from there. So finally, let me say a big thank you to the Biocytogen discovery teams that provided the data that I talked to you about today. And thank you very much for your attention. And if we still have time, I would be happy to answer any questions you might have. Okay, thank you.
Thank you for your time, and thank you for joining this webinar by Biocytogen. That's all for the presentation by Dr. Frank. If you have any question to us, please drop us an email at info@biocytogen.com.cn, and our representatives will reply you shortly. You may also scan this QR code to know more about Biocytogen from our official website. Thank you once again, and I hope to see you next time. Goodbye.